The interior of a mammal contains high concentrations of nutrients, as well as suitable and stable conditions, but the body is not easy to conquer. All the extracellular fluid is toxic to microbes. Mannose-binding proteins immediately attack by binding to the bacteria's lipopolysaccharide layer. Then they start hydrolyzing component 4 and component 2 proteins, and the complement pathway cascade begins. The bacteria's surface quickly gets covered with component 5b proteins, and each of them binds to other proteins to form a pore protein complex called membrane attack complex, and it literally forms holes on the lipid bilayer of pathogens, a powerful molecular attack. The bacteria's proteins, molecules, and ions spill out, destabilizing the intracellular chemical balance they maintain, slowly weakening them until they lose their magical chemical equilibrium and stop living. Mast cells finally arrive at the injured site and are activated by components 5A and IL-33. Activated mast cells secrete large amounts of histamine, which binds with receptors on endothelial cells, stimulating an increase in blood flow, increased vascular permeability, and increased binding of phagocytes to endothelial cells. Blood plasma leaks out, which contains a high concentration of complement components and immunoglobulins, making it harder for bacteria to keep alive. The complement proteins are very effective at killing bacteria, but one species called Staphylococcus aureus managed to survive. Its success comes from the highly defensive cell wall structure. It only has one lipid bilayer, but a thick peptidoglycan layer, and a slimy polysaccharide layer. These two layers make the bacteria almost immune to components 5b. First, it must pass through the polysaccharide layer, which it's unlikely, and then manage to pass through the peptidoglycan layer, which it's more improbable, so membrane attack complexes are almost useless.